Hello everybody, this is Javier Puy. In this presentation I will be talking about the project Global Variation in Clonality in Plantago, lanceolata. First of all, I want to thank Stephanie, Annabel, Glenda and Yvonne because they were the precursors of this project, and of course to all participants to have contributed sending samples. And first of all, what is clonality? Clonality is the ability of a genetic individual, so what is called generally a genet, to produce more than one physically independent routine unit, RAMET, by means of vegetative growth. Although there is general consensus among researchers that clonality really matters, and indeed there's increasingly a knowledge of the importance of clonality in different fields of ecology and evolution, like population biology or invasion biology, it has been actively overlooked. And just very few people have really taken the effort to open that black box. Probably because the complexity of defining individuals or because the lack of methodological approaches, but what is true is that there is a delay in the integration of this kind of important plant trait. And we have to think that it's very important. Why? Because we have to take into account that clonality it's found in the great majority of the plant kingdom. Some people say yes, that is like 80% of the angiosperms. So, and although our species doesn't regularly multiply clonally, it might have the potential to do it so. So that's why it's important to consider clonality. So paying attention to these claims for the necessity to integrate clonality because there are various effects on population dynamics. In this project, we feel brave enough to open this kind of worms or this Pandora box. So to be able to answer the following questions. First of all, we want to check whether clonality affects or is affected by underlying demographic processes. Then second, very related to this is whether it's important to integrate clonality for making predictions about population performance, so if it's necessary for ecological modeling. And last but not least, which environment biotic conditions influence clonality. So check how is the clonality distributed along environmental and biotic gradients. So regarding the first objective, I will introduce some of the processes that might be affected by the level of clonality in our populations. So just think of two populations: one that is fully sexual and one that is has potential clonal reproduction. So the first process that is greatly affected by clonality is reproduction. Although these two populations might have the same outcome, like 5 individual offspring, they are coming from different reproductive pathways. In this project, we want to check how is the relationship between clonal and sexual reproduction, whether there is an inverse or not. And also we want to check whether clonality is stage dependent, whether like some adult plants really only adult plants are having this clonal strategy, and also like to check which external factors are affecting this clonality. This is regarding the reproduction, but also it might affect the progeny establishment. Because the horizontal resource sharing between genet and their and ramets, there is some parental support. Just imagine like some disturbance coming. There is higher chance of clonal offspring to establish and survive. And of course, if this genet is the study object, we can see that being clonal, it increased the lifespan and decreased the mortality risk by having multiple lifelines. We can see in this example, if we kill them, we see that yes, only in this second case, the genet is still present. this also alters some ecological and evolutionary processes. For example, this lateral growth can benefit the populations for their competitive ability against neighbor species, but also it might affect the genetic diversity of the population. Although this is not completely clear from literature, because it's not that clear that clonality might decrease the population genetic diversity. Since new individuals in the population of clonal species may arise from this generative reproduction and clonal growth, the incorporation of plant clonality into population models is challenging, in many species. Some demographers have used several approaches for incorporating this clonal growth into MPMS, but some others and the majority consider clonality kind of negligible. So in any case, whether considering daughter ramets as part of the maternal ramet size or as a new demographic individuals, it highly affects populated life history traits. Yes, from this paper from Janowski, it shows that indeed, like some life history traits, as for example generation time, the value really depends whether we account for clonality or not. So this is what we want to check in our Plantago lanceolata, because in this article, Janowski, the authors suggest that for species that rarely exhibit clonality, it can be treated as a growth of the maternal ramet. So we really want to really test this in Plantago lanceolata to really see if this is the case. The lack of the methodological approaches is one of the causes that has created the problem of a poor integration of clonality in population demography. And why? Because it's kind of complicated to assess the standing level of clonality in a population, right? If we go one day, and we cannot estimate this. However, the development and price drop of molecular analysis, it has allowed progress in this area in recent years, and indeed this is the method that we are going to use to assess clonality in our populations. So in our case, we are going to examine kinship coefficients between pair of rosettes by genotyping by sequences using DART-seq technology. And rosettes will be considered clones when they have more than 90% matching genotype and a more than 0.45 kinship coefficient. Another situation in which the integration of clonality for ecological modeling will be necessary if it's in case that clonality is not equally distributed globally, right? So, and it cannot be predicted from macroecological factors. So one of the important objectives of this study is to test how clonality is globally distributed. So from what we know, clonality is not equally distributed around the globe, at least when we check the proportion of clonal species and their different ecological gradients. So in this example, they show that there is a more abundance of clonal species in higher latitudes and altitude. And however, if we zoom in and we check whether this is true within species clade, this is not that obvious. For example, one example is the order where our Plantago belongs to. It has more unimodal shape rather than this linear shape. And of course, similar patterns seems to happen under different gradients. But the question here is like, okay, in our project we are going to check this within species. When we check within a species, what is clear is that the number of studies that directly estimate clonality level in populations and along ecological gradients are very low, and even less when we exclude studies working on like highly clonal species or like aquatic species, or species that that accompany changes in ploidity along the environmental gradients. For the ones that are not very familiar with the topic, polyploidity is often related with self-compatible and clonal plants. So that's why. However, some studies have found that there is an increase on clonality with lower temperatures and higher precipitations, also with altitude or probability of disturbance and competition for light. However, what is clear is that our study in Plantago and lanceolata will be unique in terms that it explores clonality in an unprecedented global scale and with a cosmopolitan species. And most importantly is that it's not extremely clonal. It's not extremely clonal species. And of course, we have a great detailed demographic data. To finish, I will present you our data from our 64 sites of the net. 25 sites agreed to perform this clonality protocol. From these 25, 19 have this genomic data from the Annabelle paper, and only 5 are in non-native sites, but we don't care much because, as you can see in the map, our sites are relatively well distributed along around the world and also along genomic clusters. It's funny because sometimes there is like few points, like similar points in similar kind of sites, so it will be nice to have replicates in every site. So unfortunately, since we have experienced some delays on the project, there are no results yet to be presented, so I will show you just the preliminary ones that some of you might know already. So preliminary data shows that some extremely high variation in the rates of clonality, ranging from 91% in Ireland, while in Australia they have like 3 to 24% of clonality rate. So now the question that we make to ourselves is whether environmental seasonality is decreasing clonality and increasing genetic diversity. However, this answer and hopefully more will come very soon. Thank you all for your attention, and of course to all participants in the add-on and to funders. Thank you very much.